Hey everyone, and welcome to The Explainer. Today, we're going to explore a really fascinating technological shift that is absolutely revolutionizing the way we understand and manage our planet's soil. Whether you're an undergrad studying agronomy, environmental science, or honestly, just someone curious about the future of our food supply, you are definitely in the right place. We're going to look at how shining a simple flash of light onto a handful of dirt is completely transforming agricultural science.
Okay, let's dive into this. Here is our roadmap for today. 1. The soil analysis problem. 2. What is soil spectroscopy? 3. Reading the soil's fingerprint. 4. From labs to the field. 5. The moisture challenge. And finally, 6. The future of farming.
Kicking things off with Section 1, the soil analysis problem.
So to really understand why a new technology is such a breakthrough, we first have to understand the bottleneck it's replacing, right? Soil data is the absolute foundation of global agriculture and environmental monitoring. But getting that data, historically, it's been a massive headache. For decades, the gold standard has been traditional wet chemistry. I mean, testing for soil protein or nitrogen using something like the Kjeldahl method involves a multi-hour digestion process using literally boiling concentrated sulfuric acid. It's hazardous, it completely destroys the sample, it generates toxic waste, and you need highly trained chemists just to pull it off. Now, contrast that with modern spectroscopy. It's a clean, non-destructive 60-second push-button analysis. You simply shine a light on the soil and you're done. Understanding this stark contrast is absolutely vital for any student entering the field today.
Moving right along to Section 2, what is soil spectroscopy?
Now that we see the why, let's transition into the actual science behind this modern solution. Think of this definition as a foundational mental anchor, kind of like a flashcard for your studies. Diffuse reflectance spectroscopy, or DRS, is the measurement of how light from the visible and infrared spectrum is absorbed and reflected by a soil surface to determine its mineral and organic composition. So let's break down what that actually looks like in practice.
As humans, we rely entirely on the visible light spectrum, the standard rainbow of colors. But visible light is really just a tiny fraction of the whole story. Spectroscopy utilizes not just visible light, but the near-infrared, or NIR, and mid-infrared, or MIR regions. These infrared wavelengths allow our sensors to literally see way beyond human capabilities, peering straight into the molecular structure of the soil itself.
Now, what's really interesting about this slide is exactly what that sensor sees. Instead of pretty colors, the spectrometer produces a graph of absorbance across different wavelengths. Notice those specific dips and peaks? They aren't just random noise. They represent specific molecular bonds, like carbon-hydrogen or oxygen-hydrogen bonds, absorbing the light. This entire graph acts exactly like a molecular barcode, or a totally unique fingerprint for that specific soil sample.
Which brings us to Section 3, reading the soil's fingerprint.
We know the spectrum exists, sure, but how do we actually translate that squiggly line on a graph into usable agricultural data? Well, it all comes down to quantum mechanics. Chemical bonds inside the soil's organic matter and minerals are constantly vibrating. They're stretching, bending, and twisting at very specific frequencies. When we hit them with light energy that perfectly matches their unique vibration, they absorb it. By measuring exactly which wavelengths of light bounce back and which ones get absorbed, we can identify exactly which molecules are present. It's like a microscopic dance.
The highly complex world of chemometrics really just boils down to these 3 super relatable steps. Step 1. We shine a broad spectrum light onto our soil sample. Step 2. The spectrometer measures the reflected light, creating that fingerprint graph we just talked about. And Step 3. We feed that massive amount of spectral data straight into machine learning algorithms. The AI compares our brand new scan against huge global libraries of known soils to instantly predict what's in our sample.
The absolute game changer here is the sheer efficiency of this process. Instead of running 5 completely different, highly expensive chemical lab tests to find out the soil organic carbon, the nitrogen, the texture, the pH, and the cation exchange capacity, we get all of these crucial agricultural data points simultaneously, and we get them from just one single flash of light. It's a massive multiplier for both research and farming efficiency.
But, this naturally leads us to a massive question. If this technology is so fast, so clean, and works so flawlessly in the perfectly controlled environment of a laboratory, can we take it outside? Can we literally put it directly into the hands of the farmers?
Let's explore that in Section 4, from labs to the field.
This is where the engineering evolution of the hardware becomes just as important as the chemistry itself. We're currently living through a major hardware transition. On one side, we have traditional benchtop spectrometers. They can cost hundreds of thousands of dollars, they sit stationary in a lab, and they require the soil to be perfectly dried and sieved. Obviously, they offer incredible accuracy. But on the other side, we have the new reality: portable, miniaturized field scanners. They are cost-effective, they literally fit in a pocket, and they require minimal sample prep. And yeah, while they might have a slightly narrower spectral range, they allow a farmer or an ag student to walk a field and rapidly map out spatial data in real time. And this brilliantly illustrates how we actually got here. It's the convergence of three major tech trends: the miniaturization of optical sensors, the explosion of big data and open soil spectral libraries, and of course the massive leap in artificial intelligence and machine learning. Together, they've taken spectroscopy off the sterile lab bench and put it right onto the tractor.
But it's not without its hurdles. Section 5, the moisture challenge.
As any science student knows, no technology is just pure magic. And translating a precision lab technique to the rugged outdoors comes with some serious scientific hurdles. Taking a scanner into a real agricultural field is vastly more complicated than analyzing a dried, ground-up powder in a pristine lab. Out in the real world, you're battling variables. You have vegetation cover, where dead leaves are mixing with the soil signal. You have surface roughness, scattering the light all over the place. You have wildly inconsistent particle sizes, from fine dust all the way to chunky clay. But the absolute biggest hurdle on this entire list: variable soil moisture. See, water is a highly polar, spectrally active molecule. Because water so profoundly dominates the near-infrared spectrum, especially around the 1400 and 1900 nanometer marks, wet soil acts like a giant spectral mask. It essentially swallows the light, hiding the much weaker signatures of things like carbon, nitrogen, and other crucial minerals. Without really advanced mathematical corrections to digitally remove the water from the data, scanning wet, muddy soil drastically reduces the accuracy of our machine learning models. Overcoming this is pretty much the current frontier of soil science research right now.
Which brings us to the big picture. Section 6, the future of farming.
We've covered the chemistry, the hardware, and the challenges. So why does all this actually matter for you and for the world? Well, because cheap, instant soil data unlocks the entire field of precision agriculture. Farmers can apply the exact amount of fertilizer needed millimeter by millimeter, significantly reducing toxic chemical runoff. Scientists around the world are collaborating to build massive open soil spectral libraries to standardize global data. We can create continuous digital soil maps, and crucially, by accurately predicting soil organic carbon, we can track exactly how much carbon our farms are actively pulling out of the atmosphere, which makes soil management a key weapon in fighting climate change.
I want to leave you with this final, provocative thought to chew on. Could rapid, push-button soil data be our greatest weapon in securing global food supplies and mitigating climate change? By replacing hazardous, toxic chemicals with the simple, elegant power of light, we are illuminating a path to a much more sustainable, data-driven future. It is a really incredible time to be studying this field. Thank you so much for joining me for this explainer. Keep asking those great questions, and I'll catch you in the next one.